How are you, my dear students? Good day once again. Welcome to class of Biochemistry 407, Lecture Series 6. The topic is Biotransformation or Metabolism of Drugs. At the end of today's lecture, you should be able to define biotransformation and its role in drug pharmacokinetics. Differentiate between phase 1 and phase 2 of biotransformation reactions. Identify key enzymes such as Cytochrome P450 and their role in metabolism. Then discuss or familiarize yourself with factors affecting biotransformation. The question is, what is biotransformation of drugs? Biotransformation of drugs simply refer to the alteration of drugs that enhances its water solubility for easy excretion from the body. Where does it take place? The site of biotransformation reaction are majorly the liver, kidney, intestines, and plasma. What are the consequences of biotransformation reactions? They can inactivate drug action or activate a prodrug to become active. Now, let's look at phases of biotransformation. As shown here, you can see that there are just two, the phase 1 and phase 2. Phase 1 is the functionalization reactions, while phase 2 is the conjugation reactions. Let's proceed by looking at the functionalization reaction, which is phase 1. What is the purpose? The purpose of phase 1 reaction is to introduce a polar functional group such as the hydroxyl, the imino or imine, through oxidation, reduction, or hydrolytic reactions. Key enzymes involved are the cytochrome P450, which we shall come to talk about in details a few minutes from now. Now, this cytochrome P450 oxidizes 75% of the drugs we presently have through their hydrolytic enzymes and breakage of ester bonds, as we find in aspirin being converted to salicylic acid and acetic acid. The outcome of the reaction is to increase polarity for excretion. Let's look at example of ester bond breakage. Now, the reaction of acetyl salicylic acid can exemplify this. As I said earlier on, you have acetyl salicylic acid esterase acting on acetyl salicylic acid. Another name for that is aspirin. And of course, we have salicylic acid and acetic acid. Take note of the structure and the enzyme. Glucuronidation reaction. This reaction is essential in the process of converting a semi-soluble or partially soluble reaction to a highly soluble substances for their excretion. This process plays a significant role in drug metabolism and detoxification. It typically involves the addition of glucuronic acid molecule to a substrate. And of course, this reaction is catalyzed by uridyl diphosphate gluconosyl transferase, UGT or UGTS for plural. Through this process, lipophilic compounds are rendered more polar, which allows them to dissolve in water, making it easier for the kidneys to filter and excrete them through urine or bile. The process of glucuronidation involves two main pathways or two main stages. Stage 1 is the synthesis of the activated glucuronide donor, which is UDP glucuronic acid, and the stage 2 is conjugation of the substrate with glucuronic acid. Now let's look at the stages. Synthesis of the activated glucuronide. Synthesis of the activated glucuronide, which is stage 1. Now this reaction involves two steps. The formation of UDP glucose and the formation of UDP glucuronic acid. The glucose 1-phosphate can come from the glucose pathway. This stage creates essential donor molecule UDP-GA, that is UDP glucuronic acid, which carries the glucuronic acid moiety in an activated form. The second stage of the reaction is substrate recognition and product. As indicated here, the substrate denotes, or the substrate can be denoted or is denoted by arrow XH. X can be oxygen, nitrogen, sulfur, carboxylic acid group, as the case may be, or even a thiol. Now that reacts with UDP-GA, that is UDP glucuronic acid, in the presence of the enzyme, to form substrate glucuronide. The product is the glucuronide conjugate. Typically, the product is the glucuronide conjugate, typically larger than the original substrate. It is more water soluble due to the added carboxyl or hydroxyl groups to the glucuronic acid moiety. Please remember that we are in functionalization reaction, which constitutes the phase 1 reaction we are dealing with. Now, sulfation is a key aspect. It's a key aspect of the biochemical process where a sulfate group is added to another molecule or is added to a substrate. It is involved in many parts of biochemical process, especially detoxification, hormone regulation, and the structure of connective tissues. Sulfation modifies the function, activity, stability, or localization of the target molecule. Specialized enzymes called sulfotransferases catalyze the transfer of the sulfate group from PAP PAPs to the substrate. Now, the donation of sulfate group by PAPs is indicated here before you. Substrate XH with PAPs. Now, this reaction is catalyzed by sulfotransferase to give you the product and PAP. The first two reactions describe the two-step enzymatic process of PAPs from ATP and inorganic phosphate. And the third reaction shows how PAPs is used as a donor to other molecules. Still on functionalization reactions. Methylation. The metabolites formed are not polar or water soluble. The metabolites, in most cases, have equal or greater pharmacological activity than the parent drug. So, in essence, this reaction is used mainly for activation purposes. Now, you can see the reaction involving methylation. Few important methyltransferases that catalyze methylation of drugs or xenobiotics are 1. Catechol O-methyltransferase, phenyl O-methyltransferase, phenyl ethanolamine N-methyltransferase, non-specific transferases. Now, we have examples of substrates methylation. We have O-methylation, N-methylation, and S-methylation. The methylation for morphine, catechol, and N-methylation for primary aliphatic amines, as well as S-methylation for thiols. Presented to you here is the major pathway for biotransformation. It involves the phases, reaction types, key enzymes, substrates, and outcome. In the phases, we have phase 1 and phase 2. Reaction type, we have oxidation, hydrolysis, glucuronidation, sulfation, and methylation. Key enzymes involved: Cytochrome P450 enzymes, esterases, uridine glucosyltransferases, sulfotransferases, and methyltransferases. Substrates for phase 1 reactions, many drugs. Phase 2, we have phenols, alcohol, and acid. Secondary and tertiary amines. Then the outcomes is to improved solubility for easy excretion. Now, conclusion of lecture 6. We have seen that biotransformation is essential for drug elimination, involving a sequence of enzyme-catalyzed reaction that increased their solubility and facilitate excretion. The process is broadly divided into two, that is, phase 1 and phase 2, with each distinct mechanism and outcomes, such that it is influenced by enzyme specificity and substrate structure. To test your understanding of what have been presented, you may wish to attempt these 6 questions. 1. You can define biotransformation and explain its significance in drug pharmacokinetics. 2. Differentiate between phase 1 and phase 2 reaction in drug metabolism, including their main purposes and outcome. 3. Describe the role of Cytochrome P450 enzyme in phase 1 reaction and provide an example of a reaction they catalyze. 4. Outline the process of glucuronidation, including the key enzymes involved in the types of substrates that undergo this reaction. Explain the synthesis and role of PABs in sulfation reaction, and identify the type of enzyme that catalyzes the transfer of the sulfate group. Finally, discuss the impact of methylation on drug metabolites in terms of solubility and pharmacological activity, and name at least 2 important methyltransferases enzymes. This is the end of the lecture. Thank you for your time and for watching. My dear students, I want to welcome you from the interlude. I hope you had enough rest. We shall continue with our lecture series 7, and the topic is nomenclature, overall reaction of cytochrome P450, and multiple forms of electron transport system. Our objectives will be as follows: to understand the nomenclature system for cytochrome P450 enzymes. Secondly, to explain the overall reaction and catalytic cycle of cytochrome P450. Thirdly, describe the structure and function of this electron transport chain. Fourthly, compare different forms of electron transport system or chains, and understand the role of cytochromes in metabolic processes. What are cytochrome P450? Cytochrome P450 refers to a heme containing large superfamily of enzymes, mostly or formerly known as monooxygenases, that play a central role in the metabolism of a variety of endogenous and exogenous compounds, and are named for their unique spectrophotometric absorption peak, that is P, at 450. Absorption peak P at 450. That is, they absorb light at 450 nanometers. What are then these monooxygenases? They are a class of enzyme that catalyze the addition of a single atom of molecular oxygen into an organic substrate, while the other atom of oxygen is reduced to water. This reaction is crucial in various metabolic pathways, including that of drugs, hormones, and fatty acid, as well as in environmental processes like remediation. How are Cytochrome P450 named? The system of nomenclature for cytochrome P450 follows a pattern. For example, you have here CYP2C8. The CYP designates the superfamily. The next Arabic number indicates the gene family. The following capital letter denotes the subfamily, while the next numeral identifies the individual gene. That is the basic nomenclature for cytochrome P450. Now, look at this example. CYP2E1. CYP is a cytochrome P450 enzyme. In other words, CYP means cytochrome P450. The 2 there identifies the family. The E identifies the subfamily, and 1 indicates the specific gene within that subfamily. There are formatting conventions for cytochrome P450 enzymes. For example, when you have cytochrome P450 nomenclature written in italics, it indicates the genes, while the one or the nomenclature that is not italicized refers to the enzymes. It is of note that the superfamily is extensive, as there are more than 270 different types of gene families existing in cytochromes, with 18 found in mammals. The human genome contains 57 cytochrome P450 genes and 33 pseudogenes across 18 families and 42 subfamilies. Plants have substantially more cytochrome P450 genes compared to humans. We can now look at the overall reaction and catalytic cycle of cytochrome P450. The basic reaction catalyzed by the enzymes is oxygenation, as earlier mentioned. The enzyme incorporates 1 atom of molecular oxygen into substrates, while the other is used to form water. The overall reaction requires reducing equivalents, usually supplied by NADPH and a proton. Now, this is the reaction, the stoichiometry of the reaction. Where ROH defines the substrate, of course, O2, molecular oxygen, NADPH, the electron donor, H plus, a proton from the environment, OH, the hydroxylated product, and water formed from the reaction, of course, the oxidized form of NADPH. How about the catalytic cycle of cytochrome P450? Now, the reaction shown in the last slide, the last one before now, we can see that ROH was converted to ROOH, which is hydroxylated and more soluble than the parent compound. The cycle shows the catalytic step that is used to achieve that reaction, with ion playing a very active role in the process, in addition to NADPH. Now, the summary of this reaction, we can see that the heme ion is central to catalysis, circling between ion 2 and ion 3. NADPH is required as a source of reducing equivalent. Oxygen activation splits the molecular oxygen, incorporating one atom into the substrate and reducing the other to water. The highly reactive ferrioxo intermediate is responsible for substrate oxidation. The cytochrome P450 active site can accommodate a wide variety of substrates, leading to its role in metabolizing a diverse group of compounds. This cycle is fundamental to the function of cytochrome P450 enzyme in detoxification and biosynthesis. We can now look at the structure and function of electron transport chain. The question that will readily come to mind is, what is a transport chain, or what is an electron transport chain? I believe you are already familiar with this. You must have done in your lower level electron transport chain. But in summary, we can agree that electron transport chain or electron transport system is a series of protein complexes, that is complexes 1 to 4, mobile carrier molecules, namely ubiquinone and cytochrome C, embedded within a membrane. Shown in the picture here is a picture of the electron transport chain. Often they are housed in the inner mitochondrial membrane, and in eukaryotes and in prokaryotes, it is present in the plasma membrane. What is the function of this electron transport chain or system? Their core function is to harness the energy stored in electrons carried by molecules like NADH and FAD2. That is, this are involved in the synthesis of chemical energy in the form of ATP for cellular use. Having known what electron transport is and what their core function is, what are the different forms of electron transport system? Electron transport systems vary across organisms and cellular components or compartments. Electron transport systems vary across organisms and cellular compartments, as we have in mitochondrial electron transport chain, microsomal electron transport chain, prokaryotic electron transport chain, and photosynthetic electron transport chain. For the purpose of this lecture, we only concern ourselves with the first two, which are mitochondrial electron transport chain and microsomal electron transport chain. Now, this is a simplified structure of mitochondrial electron transport system. You can see the proteins involved: adenosine reductase, cytochrome B5 reductase, the iron sulfur 1, iron sulfur 2, which is the adenosine, and the cytochrome P450 head. For the microsomal cytochrome P450, this is the simplified structure with the associated proteins. Now we can do a comparison of the proteins or the two structures. First, in both cases, two electrons are transferred from NADPH and stored within the flavin protein. Cytochrome P450 has a single heme prosthetic group that accepts electron at a time. That is, it accepts an electron, one electron at a time. In the mitochondria, iron sulfur protein in adenosine play this role. In microsome, however, or microsomal cell, the FAD-FMN present in CYP plays the role of accepting electrons from NADPH. Now, we've talked much about cytochromes. What is the role of cytochromes in metabolic processes? We know that cytochromes are heme-containing proteins that play several crucial roles in metabolic processes, such as energy production, ATP, that is energy production in form of ATP, detoxification, biosynthesis of key molecules, and regulation of cell survival. Let's look at each one in turn. First, starting with electron transport and energy production. Cytochromes are key components of the mitochondrial electron transport chains, where they act as electron carrier, facilitating the production of ATP. This electron is essential for aerobic respiration and energy metabolism. In detoxification, cytochrome enzymes are primarily involved in the metabolism and detoxification of drugs and toxins. They catalyze oxidation reaction that makes these compounds more water soluble and easy to excrete. This process is vital for protecting the body from potentially harmful substances. Thirdly, synthesis of important biomolecules. The cytochrome P450 enzymes participate in the synthesis of endogenous molecules, including cholesterol, steroid hormones, and other signaling molecules. These biosynthetic pathways are essential for supporting various cellular functions. Finally, certain cytochromes, such as cytochrome C, have roles beyond metabolism. When released into the cytosol, cytochrome C can initiate intrinsic apoptosis, a programmed cell death pathway important for development and disease prevention. In conclusion, cytochrome P450 enzymes are a large superfamily of heme-containing homo-oxygenases. The catalytic cycle involves heme ion cycling between ion 3 and ion 2, activation and splitting of molecular oxygen, and thirdly, formation of reactive ferrioxole intermediate that oxidizes the substrate. In electron transport, cytochrome P450 differs. In mitochondria, electrons are transferred using the iron sulfur proteins, while in the mitochondria, it is achieved by cytochrome P450 reductase containing FAD and FMN. Cytochrome plays a key role in metabolic activities such as energy production, detoxification, biosynthesis of molecules, and regulation of cell death. Another word for programmed cell death is apoptosis. My dear students, you may wish to test your understanding of the lecture presented in lecture 7 by describing the nomenclature system for cytochrome P450 enzyme and provide an example. Write the overall catalytic reaction of cytochrome P450 and explain the role of each reactant and product. Outline the steps of the cytochrome P450 catalytic cycle, highlighting the role of the heme ion and electron donors. Compare the electron transport system involved in cytochrome P450 catalysis in mitochondria and microtomes. List and briefly explain three major metabolic roles of cytochromes in the body. Again, I refer you to this essential reference material or text. It will be of value to you. You need to consult it as often as you can. to enhance your understanding of the subjects being presented or being taught. This brings us to the end of lectures 6 and 7. Thank you very much for listening. Wishing you a very nice day ahead.